Michael Schmidt. I'm one of the moderators of This Week in Microbiology, and it's my great pleasure to be here this week at ICAAC, bringing you some of the hottest science topics that are being presented here for the first time at the, and I'm going to get this right, I swear, the Interscience Conference on Antimicrobial Agents and Chemotherapy, or ICAAC. It's a mouthful, and yesterday I muffled it a little bit, but today we're going to try to go 5 for 5 throughout our presentations. It's my great pleasure this morning to have with us, now I forgot your name.
It's all right.
No, we have Dr. William Mitchell from Vanderbilt University, who's going to share with us a really exciting topic on this new emerging virus that is coming out of the Far East, the influenza H7N9. And as many of you know, this is, of course, the new demon in the freezer or the closet that is coming out, and many folks are really concerned about it. And Dr. Mitchell, why don't you share with us the backstory on H7N9 and how you got involved in working with this and the findings of your study?
Well, part of it comes with association with Dr. Jürgen Rick and his colleagues at Kansas State, part of Homeland Security. The other aspect of this is my association with Hemispherics Biopharma, which I'm a board member, so everybody knows I have a competing interest there, and a natural alpha interferon that is really quite active against refractory genital HPV, intralesional injection. And the studies the company has been doing with various viruses with the Alpharon, this natural interferon, and the latest thing is with H7N9.
And interferon, for those that are not familiar with it, is a natural product produced in people yes. to effectively fight off. It's part of our innate immune system that is active all the time, surveilling our systems, looking to take out viruses.
We would not be able to exist without interferon.
Yeah, it's really the wonder drug of the human system in terms of keeping those nasty viruses in check. So we know oseltamivir is the wonder drug for controlling influenza. It's got some fundamental limitations. You have to take it at the onset of symptoms in order to have an effect. And it's unfortunate that oseltamivir resistance, the influenza viruses have figured out a way to become resistant to oseltamivir, also known as Tamiflu, to the general public, and it becomes resistant quite easily. And it's quite disturbing. Your abstract talks about the most recent outbreak in April of 2013, where there were well over 130 people who came down with the illness. 44 of those individuals died, so that's an extremely high death statistic. And unfortunately, that virus that was within that population, quickly resistance emerged. And so this is extremely disturbing. Can you share with us any insight that you have on your perceptions of oseltamivir resistance?
Yeah. Well, first of all, this is an avian virus. To be infected with H7N9, you're usually in contact with high concentrations of virus. You're a chicken farmer or whatever it is.
You have to be exposed to the poultry industry.
And the reason for that is receptors for the virus are avian, and they're in the lower bronchial tree.
So when you inhale the massive dose of virus, the virus has to go well down into your lung, which is different than the normal human influenza, which affects up on the top.
The upper airway.
Right.
Now, the thing that makes this dangerous is 2 mutational events.
Only 2?
Well, the most important 2.
Most important, which is always spooky.
The first is achieving a hemagglutinin, human type hemagglutinin.
So it can bind in the
So it
upper
binds in the
register.
upper airways. And this is alpha 2,6 versus the alpha 2,3 sialic acid in the lower, that's the avian, the lower pathway. This then becomes, you can then transmit this by droplets.
Oh, so it becomes
Yep.
droplet-borne.
It then becomes possible to be droplet form as we spread influenza, normal seasonal influenza. The second thing that makes it dangerous is the first line, well, there are two classes of neuraminidase, and this is, for the virus, let me go back. There are, on the surface of the virus is the hemagglutinin, and the second is the neuraminidase. To be active, it has to be attached to a receptor, internalized, and then to be released, the neuraminidase has to act for a full release of the virus.
So if it spread cell to cell and you make more virus.
Right. So if you inhibit that with, for instance, Tamiflu, you inhibit viral spread, and the influenza disease is then stopped in its tracks.
Yeah, because you have less virus, so you have less symptoms.
However, it is easy to develop neuraminidase resistance. And what we're reporting today is patient isolates from China. The first is a wild type that is fully Tamiflu sensitive. And the second is a patient isolate that is resistant, and it has a very conservative mutation from an arginine, a residue 292 in the neuraminidase, to a lysine.
So one amino acid change from an arg to a lys.
And both positively charged amino acids. That's surprising in itself, because it's such a conservative mutation. But that in itself makes it resistant not only to Tamiflu, but to zanamivir.
So the two first-line drugs that we
and
have.
peramivir. So all three of them.
So we have the triple whammy. So.
Yeah. Plus M2, it also carries the M2 ion resistant mutation that for the adamantines, or adamantane class, don't work either. So if one had a change in where it becomes a human-like
So
virus,
it becomes more easily transmitted person to person.
easily transmitted, combined with a widespread resistance to neuraminidase inhibitors, we have a big problem.
Yeah, I would say so. It will definitely.
It hasn't happened yet.
We've been very fortunate in that our colleagues in China have been really attempting to control the spread by taking out chicken populations and poultry farms that do indeed have this virus, and we learned about that at our general meeting.
But one of the difficulties, if you compare this to H5N1, H5N1 was very lethal to chickens. So it was easy to see where the problems were. This virus has low pathogenicity to birds.
So you don't have the bioassay of looking at your flock for a dead bird.
You can't just look at the flock.
You have to test the flock, which is substantially labor intensive, to bleed the chicken and ask the question, is the virus there, or do they have antibodies against the virus after having an infection? So it's a low path virus in the chickens. But when
high path for humans when they get it.
When they get it. The good news right now, so we don't start panic, is that this particular virus, H7N9, is still extremely difficult to get into people because it has to penetrate deep.
It has to go to the lower airways.
But the unfortunate thing is that this resistance emerges so quickly. So what's your headline news for today is you've actually come up with looking at our natural antivirals.
Well, this is a late breaker, and it's a late breaker because the data's just
coming
out
in.
Out of the lab. And I just would mention that the primary person doing this work in biohazard containment facilities at Kansas State is Dr. Liu, Sin Fong Liu, who has produced results really very quickly. And the bottom line is, both with the wild-type virus, both Tamiflu and the natural interferon, Alpharon, inhibit the virus approximately about the same.
Well, that's good.
And however, with the Tamiflu resistant virus carrying this R292 lysine
Substitution.
mutation, it completely wipes out. The vast majority of the activity is wiped out. However, the activity with Alpharon is even better. It wiped out 100% of the activity.
Now, unlike oseltamivir, with this interferon, you don't have to have the magic window of administration between 24 and 48 hours of symptom onset, because it's literally stopping the replication of the virus rather than the release.
Yes. Now, the earlier you get it,
the better off you're going to be, obviously.
Yes.
But
yes.
But the good news is this is already an ethical pharmaceutical.
Yes.
So we have the know-how to make it.
Yes.
The really interesting findings are that you can now use it in place of oseltamivir.
Yes.
When patients present
Yes.
with H7N9,
Yes.
you have a new class of, now this is a new class of drugs heretofore not used to treat human influenza, and this happens to be an avian influenza that is accidentally being expressed in people.
The whole point of the exercise was to prove that oseltamivir-resistant virus was still susceptible to interferon, and that I believe has been proven. So there's one other nuance with this. It's not part of the other thing that we had in the poster is not in the abstract, but I thought it was worthwhile adding, you'll see in the poster, is that the interferon, this natural interferon, surprisingly, is active in pig cells against influenza. And this comes from a number of years ago, a veterinarian was treating pig coronavirus with this particular preparation, but doing it orally. And the pig coronavirus wipes out piglets in pig farms, and it's devastating to pig producers. You don't get long-lasting immunity in the sow. And so whatever drove him to this, I don't know. But he was treating thousands of piglets with oral small doses of this. And with the highest dose, he salvaged half of them.
That's pretty incredible.
So this then led to looking at whether this could be effective in humans. And so the study was set up with H5N9, using low-dose oral. And at the highest concentration that was used, low-dose oral, in rhesus macaques challenged with H5N9, even though they would get an infection, they would produce new virus, it totally protected the lung. Well, 90% protected the lung from the destructive effects of H5N9.
So as the virus would trash the cells in its normal life, recruiting the immune response and all the evil cytokines,
But strangely enough, at that particular concentration, it did not reduce upper respiratory viral titers or the lung titers. I don't know why, but it suggests to me it with higher doses, oral. And this is just a fraction of what's normally injected, so that one could possibly use this as prophylaxis.
Well, I'm sure our prescribing colleagues already know the answer to this question, but since I'm not a prescribing colleague, I'll ask the question. How do you administer this interferon? Is it an injection? Is it a pill?
Yeah, this Alferon is like all interferons, or injectable. For the genital HPV, this is refractory. This is a intralesional injection. It works pretty good. For the low dose oral, and this is in its infancy, which I think we undoubtedly will be doing with H7N9, and also in macaques, and also it looks like we'll be able to do it in swine also. So we'll have two animals.
Well, let me ask you the hardest question to answer. Do you anticipate that the influenza virus will be again clever and become resistant to this interferon treatment?
No.
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 So an absolute from a biologist is a remarkable thing.
Well, and the reason I say that, the influenza virus is already tries to defeat interferon.
Correct.
And it does it through one of its internal proteins, inhibits the action of interferon. Now, the only way around that is to make that better. That's possible.
That's the job of the pharmaceutical industry.
That's possible. But it's not a viral target. And it's much harder to introduce mutations that are going to affect a
host response
gene. And in this particular, with the natural product, it also has 7 different species. Alpha interferon, there's 12 or 13 different gene sequences. And this is a Sendai virus induced in white blood cells. And 7 of these species are there. And it's 10 to 100 times, depending on which recombinant interferon you look at, 10 to 100 times more active than recombinant interferon.
Well, this is just tremendous. Now we're going to do our public health bit. So the question I get asked often is, given the press about H7N9, should I still get a flu vaccine?
Absolutely.
I mean, it's the best prevention.
Absolutely. Absolutely.
And many of our colleagues now out there are running flu tents where they're lining up healthcare teams and they're asking them to be vaccinated. And this year it looks like everyone's going to be using a quadrivalent vaccine. And of course that's against H1N1 and H3 and what's the neuraminidase in the H3? Is it?
I forgot.
Yeah, it's hard without a scorecard.
It changes every year.
It changes every year. So the vaccination process continues.
Well, now, at Vanderbilt, we will immunize every employee in one day. This is 10 or 12,000 people in one single day.
And we have a tremendous number of flu vaccines out there today. It's just more than the shot in the arm. You can get it nasally. So there are a lot of opportunities and a lot of different vaccines available today.
My major problem is getting my spouse and my children, who obviously now live far away from home, to go and get the influenza vaccine and get their children vaccinated.
Yeah, it's, the Centers for Disease Control and Prevention, of course, is recommending that everyone get a vaccine. I mean, it's tremendous protection for a relatively inexpensive price. And, when I am asked, how do you describe the symptoms of influenza, the common thing I always point out is, you have the flu when you're too tired to text. And every medical student I tell that to immediately appreciates what lethargy means. We have a question from the audience.
Yeah, Michael Smith, MedPage today. Dr. Mitchell, can you put a little salt on the tail of this molecule for me? This is, interferon, as you say, comes in several varieties. Is this a recombinant interferon, or is it extracted from?
There's an echo in the room, and I'm not hearing the question.
I don't know. Can anybody else fix that? If I lean back, how's that?
Well, we can try it. I think just.
Okay.
How's that?
Just repeat the question and
Sure.
I'll...
Is it better? Okay, sure. Can you hear me? The question is, can we get a little sort of definition of what this molecule is, where it comes from? Is it a you just said it was an alpha interferon.
Okay.
A little bit more about it. Where does it come from? Is it now in clinical trials in any
For influenza.
for influenza, for instance, that sort of thing?
Okay, he's specifically asking about the derivation of this interferon and what it actually is. Is it a recombinant? And has this interferon moved into getting an indication to treat H7N9 or influenza in general?
Well, probably be premature in terms of treating clinical trials, because we don't have a pandemic, but it certainly is an approved injectable, and in the event of a pandemic, I'm quite sure it would get emergency approval. And I can tell you the company has invested a lot in being able to upscale its production of this interferon.
So it's a recombinant interferon, and
no, this is a natural interferon, but it's done in vast vats rather than individual little
tissue culture wells.
Yeah. So it should be, if the occasion calls for it, should be available.
You had a follow-up?
Yeah, a couple. I also wasn't quite clear on, you said that it had little effect on the wild type, but it completely stopped replication.
No. Alpharon is, or this natural interferon, has approximately the same activity against the wild type virus as does Tamiflu. However, in the resistant virus, patient isolate, it was completely active while the Tamiflu activity was markedly reduced.
But the interferon was able to inactivate.
Was actually more active than it was in the wild type.
Thank you.
And if any of you in the Twitterverse have a question, you can post your question using the hashtag ICAAC. Any other questions, Jim, from the Twitterverse? Hearing none, I think we're done, Dr. Mitchell, and I thank you for your time, and I look forward to seeing your poster, and it's down on the floor today, so you can brush down there.
I actually put it up.
Before you came up?
Yep.
Yeah, so we're looking forward to going down there and wandering the halls and taking a look at it. So thank you very much for your time, and best of luck, and thank you for your work.
Okay, thank you.
Thank you.
Explore the fundamental role of microbes in the natural history of our planet with Microbes in Evolution, the world that Darwin never saw. Available at estore.asm.org.